All right. So being that this all focuses on ribulose 1,5-bisphosphate in the Calvin cycle and it being regenerated, so it's probably a 5-carbon sugar worth knowing the structure of. So I would think that's probably pretty important. This is the oxygenase activity of RuBisCo. So RuBisCo is supposed to, fix carbon dioxide, combine it with ribulose 1,5-bisphosphate, and make 2 glyceraldehyde 3-phosphates. So unfortunately, that might not be the only result. In this case, RuBisCo might react with oxygen instead of carbon dioxide, producing glyceraldehyde 3-phosphate, but then also producing, bless you, 2-phosphoglycolate instead. So 2-phosphoglycolate. So in this case, we're losing out. Our goal was hopefully to make 2 glyceraldehyde 3-phosphates. We only get 1. And we get this 2-phosphoglycolate instead with the oxygenase activity. So there's a process called photorespiration, and the whole goal is to recover this 2-phosphoglycolate. It'd be nice if we could use those 2 carbons and not just complete waste. And photorespiration allows us to take this 2-phosphoglycolate that is waste produced by RuBisCo's oxygenase activity and turn it back into glyceraldehyde 3-phosphate. So if RuBisCo uses O2, and again, this is a complete waste from what we can tell, but if it happens, every oxygen molecule RuBisCo uses, you lose a carbon dioxide, and you're going to use up an extra ATP in the process. Question?
Does it have higher affinity for carbon dioxide or oxygen?
It has a higher affinity for carbon dioxide, but what's there more of in the air? The atmosphere. There's more oxygen, so it's kind of an interplay. So it definitely has a higher affinity for a carbon dioxide, but because there's greater concentration of O2 in the air usually, it's kind of this problem. So as CO2 concentrations get lower and O2 concentrations get higher, this gets to be a bigger deal. So we'll see some plants have some interesting ways to get around this issue. But again, we can really find no reason for RuBisCo, this being favorable in any way. It just looks like it's just part of the way of RuBisCo functioning as a CO2 fixer. This also comes along for the ride. So C4 plants. So everything we've talked about till now with the Calvin cycle was dealing with C3 plants. And the whole goal of the Calvin cycle is to make a 3-carbon chain glyceraldehyde 3-phosphate. So C4 plants kind of do something interesting. So they're looking at making 4-carbon sugars instead. So and it turns out it's more costly to make 4-carbon sugars than it is 3-carbon sugars. So, and this actually is supposed to benefit them in some way. So we got to explain this away, because if I'm going to make a 4-carbon sugar and a 3-carbon sugar, okay, great, who cares? But if it's going to cost more energy per carbon, then there better be a reason we're doing this. So in this case, so carbon dioxide is actually turned into bicarbonate, combines with water. So, and PEP, phosphoenol pyruvate carboxylase, so is what actually combines phosphoenol pyruvate with the 1 carbon, in this case, bicarbonate to oxaloacetate. So anybody remember how many carbons PEP had? 3. He had 3. Notice phosphoenol pyruvate. Next steps turned into pyruvate, and they both have 3 carbons. So in this case, phosphoenol pyruvate has 3 carbons. We're going to combine it with bicarbonate, which has 1 carbon, and we form oxaloacetate, which has 4 carbons. 4 carbons. So this is our C4. We just made a 4-carbon sugar. We have fixed a carbon here, so, but not in the form of carbon dioxide, at least not immediately. It was actually in the form of bicarbonate. So what's nice here is that your PEP carboxylase is not actually binding carbon dioxide, it's binding bicarbonate. And it has 0 affinity for oxygen. So we're not relying on RuBisCo in this case to actually do the carbon fixing. We're relying on PEP carboxylase. And PEP carboxylase has no oxygenase activity. So we don't have that whole wasteful process. So in this case, even though this costs a little more, so we're avoiding some problems that RuBisCo has by doing it. So kind of funky the way this works too. The oxaloacetate is then going to be converted into malate. The malate is going to be transmitted through the plant cells are connected by channels called plasmodesmata. So it's going to be transmitted through from the mesophyll cell to the bundle sheath cell. So, and here the malate is going to turn into pyruvate, go through the cycle. Pyruvate is going to go back over to the mesophyll cell, turn back into phosphoenol pyruvate, using up a couple of ATP in the process. So we use up some NADPH, we use up some ATP along the way. in the process here, the NADPH gets regenerated, but the ATP just gets used. It's going to cost 2 extra ATPs to do this. What's nice, though, is essentially what I've done is I've taken a way of fixing carbon in the mesophyll cells, but then where's that carbon come back out? Over here in the bundle sheath cells. In what form? CO2. And it's going to go through the Calvin cycle. Why didn't we just do the Calvin cycle to begin with then? So, well, make a 3-carbon sugar, but the idea too is that now, if I keep doing this enough, I'll start making CO2 more and more concentrated for the bundle sheath cells, higher than normal concentrations you'd find in air. Why is that a good thing? It'll limit RuBisCo's oxygenase activity. So these CO2 here at the bundle sheath cells is going to actually be used for the regular Calvin cycle. The idea, though, is that now because there's more CO2 than normal around, I don't get a whole lot of oxygenase activity, so I don't get a whole lot of that RuBisCo waste activity going on. So it costs a little more money to be a C4 plant, a little more money, a little more energy, a little more ATP initially, but hopefully we more than make up for it by not having wasteful RuBisCo activity, or at least not as much anyways. Cool, your C4 plants are classically tropical plants, by the way. Although corn's your classic example, and I don't know how tropical that is, but anyways. Cam plants, another interesting way, and these are more like your desert climate plants, which hopefully you've seen a few lately. And the idea is this, so desert plants, what are they not getting a whole lot of oftentimes? Water, so they want to conserve water loss. So in this case, to absorb carbon dioxide, they have little pores in leaves called stomata, and they got to open those up in order to absorb carbon dioxide. So, but when they open those up, what might they be losing? So water. Same thing, your lungs. When you want to absorb oxygen, you have to open up your lungs, and your lungs are a big way for you to lose water all the time. So same problem in plants. When they're trying to get carbon dioxide, though, that's when they might be losing water. So if you look, anybody fill up your gas tank lately? Cool. Was it painful? Yes, it was. In the middle of summer, when do they recommend you fill up your gas tank? Early morning, late at night. Why? So it's cooler, and why is that a good thing? Less gasoline evaporates away as you fill up your tank. So one, you're getting more gasoline for your money's worth, and you're not polluting the air. But the idea is similar here. So at night, if they open up their stomata, preferably at night, these desert plants, to take up CO2, that's when they're going to lose the least amount of water to evaporation. And so that cam plants only open their stomata at night. That's when they absorb carbon dioxide. They store that carbon dioxide as malate. And then in the morning, when there's plenty of light, they turn that malate back into carbon dioxide and use it for the Calvin cycle.